The overall goal of this procedure is to image an embryonic cochlea taken from a fluorescent reporter mouse at 30-minute intervals over 5 days. This is accomplished by first harvesting embryonic day 13 cochleae from fluorescent reporter mice. Next, the cochleae are cultured in vitro. Then, the cochlear explant cultures are placed in an incubator microscope. Finally, an automated imaging routine is set up, and the cochleae are imaged. Ultimately, long-term time-lapse imaging of cochlear explant cultures can be used to show morphogenetic movements of the cochlear epithelium as a whole, as well as changes in reporter gene expression over time and the effects of slow-acting pharmacological agents.
The main advantage of this technique over existing methods like manually photographing explant cultures over a time course is that this system is entirely automated. It allows more time points to be collected, and it ensures that the same region of the cochlea or set of cells is imaged at the same orientation at every time point, all while growing in a regular tissue culture incubator.
After supplementing Dulbecco's modified Eagle medium, or DMEM, and Hank's balanced salt solution, or HBSS, according to the accompanying text protocol, under a laminar flow hood, use 5 milliliters of unsupplemented DMEM to resuspend 200 microliters of basement membrane substrate. Pipette 150 microliters into the center of each 10 mm well of a 35 mm diameter glass-bottomed culture dish. Incubate for 40 minutes in a CO2 incubator at 35 degrees Celsius. To prepare the workstation and tools, turn on the laminar flow clean bench and use 70% ethanol to spray it down and to soak forceps, spoons, and a black silicone-coated dish. In the clean workstation, harvest embryos of the appropriate gestation in ice-cold HBSS, supplemented with 1% heaps. Using a fluorescent stereo microscope, identify embryos that exhibit reporter activity externally or within a visible organ and transfer them to fresh, ice-cold HBSS with 1% heaps. For collecting temporal bones, using a cool light source and fine forceps, work quickly to clip at the cervical vertebrae and below the jaw to collect the heads. After collecting the embryo heads according to the text protocol, carefully open the skull. Then, remove the brain, trim off the front of the head, and transfer the posterior skull to a dish with fresh ice-cold HBSS heaps. Carefully dissect out the peanut-shaped temporal bones, taking care to keep the vestibular system intact. To dissect the cochlea, transfer the temporal bones to a black silicone elastomer-coated dish in ice-cold HBSS heaps. Pin the vestibular region of the bone by inserting pins at an oblique angle in order to stabilize the temporal bone and create room for the forceps. Next, carefully remove the cartilage surrounding the cochlea by inserting one tine of the forceps into the cartilage at the outer edge of the base of the cochlea and clipping a hole in the cartilage. Clip horizontally across the top and diagonally, and carefully lift off the front section of the capsule. Insert a prong of the forceps in between the remaining cartilage and the duct, and gently clip off the remaining cartilage. The apical surface of the cochlea is now exposed. Starting at the base, catch the area where the roof of the duct meets the cochlear epithelium and open the cochlear duct. Gently peel off the roof, trimming when necessary, until it is completely removed. Trim off any portions of membrane left on the medial side of the duct. Then, clean the duct of excess mesenchymal tissue and detach the duct from the vestibular system. To culture explants, place one luminal surface up in the center of a substrate-coated glass bottom culture dish. Carefully draw off the liquid and leave for 2 minutes. Then, dropwise, add 150 microliters of supplemented DMEM to the explant, taking care not to disturb it so that it can settle to the substrate. Place the glass bottom dishes in a deep 12 cm diameter petri dish with a small dish of sterile water to maintain humidity. Put the cultures into a 35 degrees Celsius incubator overnight in order for the explants to attach to the substrate and flatten. To perform live imaging, after selecting samples, use a fluorescent stereo microscope to evaluate the condition of each explanted cochlea and select only explants where the duct is intact and attached to the glass from the base to apex. Set the incubator at 35 degrees Celsius with 5% CO2 to culture cochlear tissue. Gently aspirate the supplemented DMEM from the cultures and replace it with at least 500 microliters of fresh medium. In cases where explants are loosely attached, pipette a ring of medium around the edge of the dish. This extra liquid will make a miniature humidified chamber without disturbing the explant while it continues to attach to the matrix. The microscope in this example has a rotating platform that holds 8 35mm sample dishes. Under the laminar flow hood, replace the plastic lids with glass lids and insert the dishes into the sample dish holder. If it is necessary to change the media during the course of the experiment, to avoid disturbing the explant, use hinged dish covers that will allow opening of the lids while the dish stays in its fitting in the microscope. Place the sample dish holder inside the incubator microscope, taking care not to dislodge the explants from the bottom of the dish or to disturb the medium. To set up the imaging routine, switch on the microscope, UV lamp, and camera, and open the imaging software. For each sample located, pick an imaging area, plane of focus, and exposure, keeping in mind not only the view of the explant at the time of starting, but also how the tissue will move during the course of the run. In the fluorescence channel, set a Z-stack centering around the selected focal plane. Then, set the frequency and length of the sampling for the experiment. This will be limited by the time it takes to collect images. To generate a movie, at the end of a time-lapse period, use a software such as Metamorph to open the image files for a sample. Frame by frame, pick the best focal plane for showing the cell population of interest. Finally, convert the images to a .avi file, or export them as a montage to generate a set of images that can be opened and analyzed in image processing software or converted to other formats. Shown here is a movie demonstrating how an organotypic cochlear explant will grow if plated on E13.5. A SOX2 reporter mouse was used to visualize the prosensory region as seen in green. As the cells of the non-sensory regions divide and move, the non-proliferative sensory region in green undergoes tissue rearrangements and convergent extension movements. A second time-lapse experiment, centering on the midbase of a different cochlear explant, demonstrates that as it extends, the prosensory region narrows. When first in culture, the tissue thickness makes it possible to identify regions of expression, but not individual cells. However, after 3 days in culture, the tissue has flattened considerably, making it possible to visualize individual fluorescing cells.
Following this procedure, other methods like immunofluorescence staining or protein and RNA extraction can be performed in order to evaluate changes in gene expression at the molecular level. After watching this video, you should have a good understanding of how to dissect and culture cochleae and how to use an incubator microscope to perform live imaging on whole explants over a period of days.